Hello everyone, this is Dr. Lashik, and this lecture is going to cover the beginning part of Chapter 10. So we're going to talk about simple experiments. This particular lecture will cover two example experiments that we'll be referring back to when we talk about all of the important concepts in this chapter, and also experimental variables. The first experiment we're going to discuss is the seeing red study. The second experiment is going to be the eating pasta study. And so again, we're going to refer back to these two examples as we talk about each concept in this chapter. Let's start with the seeing red study. So researchers discovered that the color red is associated with things like caution, danger, and other things that we hope to avoid. So a psychologist hypothesized that if a participant or a student is presented with the color red before taking a test, that they would do more poorly compared to those who were not presented with the color red. So what they hypothesized was that the color red would make the students or participants enter what is known as an avoidance orientation. For example, avoiding failure instead of trying to succeed. So if they had not been exposed to the red color, their hypothesis was that they would not enter an avoidance orientation, but instead an approach orientation, and they would be more focused on trying to succeed rather than avoiding failure. So in their first experiment on this hypothesis, participants were requested to complete a series of anagrams, which means unscrambling words. They all received the same test booklet, but their participant ID number was printed in either red, green, or black ink inside the test booklet. What they discovered was that those who received a test booklet that had their participant ID number written in red ink ended up having poorer test scores on the anagrams, on these anagrams. They then redid the experiment several times, changing different aspects. They tried making the outside cover of the test booklet different colors. They changed it to a vocabulary test instead of an anagram test. They changed it to a math test instead of an anagram test. And all of the experiments had the same results. Those who saw the color red in any degree ended up with lower scores. And as you can see from these charts here, that demonstrates just that. So in the top left chart, the number of correct anagrams relates to the color of the ink. So the color red has the lowest bar on the bar graph. Same with when they did it with a booklet cover and color instead of the typeface of their participant ID number. And same when they did it with vocabulary or math test. The second example that we're going to discuss is the eating pasta study. So the researchers in this experiment hypothesized that the size of a bowl one takes before eating influences how much food that person will take and then subsequently eat. So the participants were asked to take a bowl. It was either a large bowl or a medium bowl. And then they were asked to get in line and serve themselves from this communal bowl of pasta, kind of like at a buffet table. The amount each participant took was then weighed, and then when they were finished eating and they were allowed to take as much time as they wanted, they had to bring up what was ever left inside their bowl, weigh it again, then the second amount was subtracted from the first amount, and they could get a number of weight of how much each participant A took and then B ate. What they found out was if a participant was given a large bowl compared to the medium bowl, the participant not only took more food, they ate more food. So now let's talk about experimental variables, and we'll reference the Seeing Red study and the bowl size study several times as we discuss these concepts. So in science, an experiment means that the researchers have manipulated one variable and measured a second. The manipulated variable is the variable that the researcher has changed in some way depending on the experimental condition. The condition is also known as the level. the manipulated variable must have more than one condition or level. For example, the manipulated variable in study 1, the Seeing Red study, was our ink color. In that first experiment, the first version of the experiment, it had 3 levels or conditions. The participant either could have been exposed to red, green, or black ink. In the second experiment, the eating pasta study, the manipulated variable was the bowl size, and it had 2 levels. Participants could have experienced a large or a medium bowl. The second type of variable is the measured variable. This is what the researcher measures and hopes changes due to the manipulation of the first variable. For example, in the Seeing Red study, they measured how many anagrams the participants solved, and they hoped that that changed due to the ink color, so due to the manipulated variable. In the second experiment, the eating pasta study, they measured how much pasta the participants ate, and they hoped that changed due to the bowl size, so what they manipulated. These are also known as independent or dependent variables. The independent variable is the manipulated variable, like ink color. The condition or level, as we just said, are synonymous with one another, can be used interchangeably. And these are the different variants of that manipulated variable. So if we have ink color as our manipulated variable, we would have 3 conditions or level: red, green, and black ink. The dependent variable is the measured variable. And we can think about this to remember the difference between dependent and independent variable. It's really easy to think about it if you talk about it as what depends on what. So the dependent variable typically depends upon the changes made to the independent variable. For example, the number of anagrams solved depended upon the ink that the participant was exposed to. And finally, control variables. In the ink color experiment, everything besides the ink color was kept exactly the same. The researchers wanted to make sure that the only difference was the ink color between each condition, so that if the results were different between each condition, they could conclude that the only possible reason was due to the ink color. All of the other aspects were held constant, and these constant aspects are considered control variables. For example, each participant couldn't have any difference with their test booklet. They had to have the same size of text. The experimenter who administered the exam had to be the same person between each participant. The exam questions all had to be exactly the same. The time limit had to be exactly the same, because anything that was changed with those other control variables, we could then say that that was what caused the difference between conditions rather than the ink color. So you have to make everything else completely constant to make sure that only the thing you changed, your independent variable, is what caused the changes of the dependent variable.